So thanks for the nice introduction. My name is Erni, and I'm heading the lab for general and theoretical engineering at the University of Duisburg-Essen. And within the next 45 minutes, I want to give a brief overview about the research we are carrying out, as indicated here, on plant and insect monitoring with terahertz waves. So let's talk first, why using terahertz waves at all? So when operating at frequencies or at that high frequencies, that allows for a radical miniaturization of even comprehensive systems like a radar system that might also include the antennas, and that on a chip, and such a chip would have a very small footprint of only a few square millimeter. And on the other hand, it's also very lightweight. That means that such tiny terahertz technologies allows for a continuous, let's say 24/7 monitoring of small local plant insect ecosystems at the high spatial resolution, and also without being detected by all the actors in such an ecosystem, namely, as shown here, the insects that are hovering, for example, above a blossom of a plant. So referring to plants, this high spatial resolution, this capability allows us to monitor internal states of plants and estimate probably also stress with respect to contamination pests. And regarding the insects, we also want to discern the players in this ecosystem, namely discern the different insects that are hovering around such a plant. And what is also an important topic is the investigation of the exposure of such insects to the surrounding electromagnetic field. That is something I will talk to the very detail. So plant and insect monitoring with terahertz waves is, and that I have to admit, an ongoing venture. So we have started, let's say, 3 years ago. And what I'm going to present is shown here in the agenda. So I will talk about plant monitoring, and in particular on heavy metal contamination in model plant leaves. I will discuss a similar topic, namely water content monitoring of geranium leaves, and then move to a sort of near field, far field scattering analysis of plant stem at these kind of frequencies in order to look into the plant stem itself. Then we will switch gears to bee monitoring, and there we will look at the analysis of terahertz bee radar cross-section that might be acquired within experiments, but also we want to look at artificial bees, so-called digital twins. And we also present some experimental setups and radar measurement setups, including a harmonic radar system for bee tracking. Then the last part of my talk will focus on bee exposure. And there we also have to look into the bee morphology itself and also into corresponding digital twins. To get reliable models, we have to look at material characterization of specific bee parts. And at the end, I will talk about this very important topic, namely the electromagnetic energy uptake and the specific absorption rate in bees. I will end my talk with a short conclusion and even with a shorter outlook. So let's start with plant monitoring. What I show here is a very successful research that has been carried out by Martin Hofmann and co-workers at the University of Bochum. They looked at terahertz detection of heavy metal uptake in plant leaves, and they looked also at a very specific plant. It's the model plant Arabidopsis halleri, which is actually a heavy metal accumulating plant. And for the analysis, they used the CV homodyne terahertz transmission spectroscopy system operating in the frequency range between 100 and 800 gigahertz. So the system is shown here on the upper right. It's actually has, let's say, 2 lasers operating at different laser frequencies. One of the lasers is tunable, so the difference frequencies, it's actually the terahertz frequency that we want to exploit here. So the optical path goes here to a pin photodiode that acts as an emitter. Then on the detector side, we have a photomixer, and the leaf is then actually put into the let's say, terahertz beam. So the leaves have to be prepared. They undergo air dry process, and they will also be pressed in order to provide a sort of regular surface. And what we see here on the lower left are actually results from a transmission measurement. And what we can already observe here is a bunch of blue spectral responses and red spectral responses. The red one stands for contaminated leaves, whereas the blue ones are the uncontaminated references. So what we can see here is that let's say the slope, so we see also that the red curves undergo a much steeper rollover, so the larger slopes of the transmission spectra somehow correlates to the contamination, here in particular of cadmium and zinc. And the question is, what is the underlying mechanism that alters the transmission in the contaminated leaves? This is an open discussion. One conjecture is that it changes the water binding features within, let's say, the plant tissue, or it also interacts with cell walls. From a more engineering perspective, we can also say it alters the complex dielectric function, namely due to the presence of ion binding. But this is, as mentioned before, still an open issue. So this methodology works pretty fine with dried leaves. On the box plot on the right-hand side, we see a different set of measurements corresponding to a different set of contaminated soils where the plant has been grown. And what is visible in all cases is that we really can classify, let's say, contaminated leaves from non-contaminated ones. So the open question is whether this also applies for fresh leaves. And here we have a strong challenge because the water signature in the leaf is actually masking or has a strong influence on let's say, the transmission. And this slope differentiation with respect to the contamination is actually lost. But what it has been said is that even though the transmission spectra show a huge variety, and it is also believed that using, for example, machine learning, one could try or even get a classification system that would successfully discern between contaminated leaves and the reference probes. Just to draw a very speculative vision, and this is not part of the research, this is what is my own idea in a certain sense, if we would succeed to look at fresh leaves and even using a more challenging measurement setup in the reflectometry, and that would mean that the reflectometry on a whole field of such plants might reveal a hidden contamination topography of the underlying soil, because these plants are actually mapping the soil contamination into their extremities, namely into the leaves, and a hovering drone could probably also reveal this sort of contamination landscape. So moving now to the next research that has been carried out in Niels Weimann's groups on terahertz monitoring of water content in geranium leaves. Here I want to start with an example of precision farming in Israel. What you see here on the right-hand side is an avocado that is somehow clamped between a very strange-looking device. I will come to that later. So I have to mention that precision agronomy is also a long-standing issue in Israel. They have also, by their national history, they are actually the inventors of this kind of droplet watering system. So in order to provide a pinpoint watering, you also need actually, let's say, you need to sense the plant state. And in this example, we see that this clamp are actually measuring the mechanical extent of the avocado. And on the other hand, they also use capacitive measurements in order to get some information about the water content and the ripeness in order to provide this very precise watering, let's say, strategy. So why starting with this? Because at terahertz frequencies, one could actually downscale the whole let's say, sensor in a certain sense to provide a very compact terahertz leaf sensor. So what they did in their group, they developed very tiny terahertz transmitters, receivers, and even transceiver chips based on resonance tunneling diet within an indium phosphide material system. So they use double barrier for, let's say, the transmitters and triple barrier for the detectors. And what I show here on the right-hand side is our first water content, is a first water content analysis on the geranium leaves using a transmission measurement in the far field, having, let's say, a voltage network analyzer as the transmitter, and the detector is realized by this RTD, triple barrier RTD, at a chopped terahertz signal of 350 gigahertz. So we see here two regions of interest, and let's say this far field scanning has also provided some topographies of water content that are just shown below. The problem here is they looked 6 days, over a period of 6 days to this leaf, so the leaves dries out, and what I show here on the right-hand side is a snapshot of the first 3 days, and what we can easily see is that the dry out of the, let's say, of the leaf. Then, just to draw a vision in connection to my introduction here, having such tiny terahertz transceivers, what one could also think of a very tiny clamp that could be applied to a plant leaf or even to an interesting part of the plant during the whole, let's say, life cycle of the plant. And that would be a very nice feature because it doesn't influence the plant at all, and it would provide a very, let's say, precise, would provide information about, let's say, the plant in this regard. So moving further, what I want to show here is something from my own group. So we are looking at the plant stem using near field and far field analysis with corresponding azimuthal scanners in order to retrieve the cross section of the plant. So the goal is to reconstruct the plant stem's cross section. Why do we want to do that? Because by solving the inverse problem, we could also estimate the cross section of the plant, and that would allow us also to look into the channel systems of the plant, because in the plant there are two sorts of channels, namely the xylem, which is responsible for the upstream, namely for the nutrients up into the extremities of the plant, and then the phloem, which is carrying the downstream, for example, glucose back to the root. That happens usually during diet. So what we will use within our pretty fast forward solver is an aggregated center T-matrix algorithm, abbreviated as RACTMA. I don't want to go too much into details here because what I can say is extremely fast. So we just to drop some numbers, we can analyze even more than 3,000 scatterers. And if the structure is introduced into the algorithm, then one can also analyze up to 10,000 excitations and detections per second. So that means actually we can generate a number of far fields or scattered fields that is, let's say, that is very useful if we let's say, analyze a tomography setup as I will show in just an instant. So the data retrieval, as mentioned before, we use an azimuthal scanner, either for the near field probes in transmission or then for the far field scattering data in order to set up radar cross sections and then to start with the reconstruction using the inverse problem. So on the upper right, we see our very preliminary near field probes made of polymer, so operating at 230 up to 330 gigahertz. These probes were not that successful because the polymer does not confine the terahertz field that much. So we have moved to our further developments of near field probes using 3D printed alumina material probes with a diameter or a cross-section of 770 times 400 microns. And one of the most important parts here is to also look at the transmission from the WR10 waveguide system into the proper probe. So these probes are actually developed for the W band operating between 75 gigahertz up to 110 gigahertz. So we are not yet at the, let's say, at the terahertz frequencies, but new developments are on the way. So one idea is also to introduce active transceivers within these probes operating at even higher frequencies. So in order to look to deal with the reconstruction of plant stem cross-section, we also use mock-ups, microfluidic mock-ups, as shown here below. So these mock-ups contains a sort of arbitrary distribution of channels that can be also filled with a liquid. And on the right-hand side, we see successful reconstructions of the cross-section, given the radar cross-section at 120 gigahertz up to 160 gigahertz. So on the next slide, I want to show such a system in action. So we will use our largest setup in order to reconstruct the stem of a bamboo. So that means it's actually a very simple cross-section, but for me, it's important then to just give an insight how this works. So I will start the video and will interrupt that. So what we see here is we will investigate a bamboo rod using a far field, let's say, scanning approach at 170 gigahertz, and we will also use the fast transfer matrix-based analysis for the reconstruction of the cross-section. So starting here with what we see here is the absorber on the transmitter side in order to prevent back reflections into the measurement system. Then here, that's the prominent appearance of the transmitter horn antenna. Here we introduced a bamboo rod, and also we have to cover the metallic part by an absorbing structure in order to prevent back reflections. Here we see the receiver antenna that will somehow circle around the bamboo rod. Here in the background, we see the transmitter antenna that illuminates the rod. So, and what we actually measure is the forward transmission factor, which is nothing else than a normalized version of the scattering amplitude. So we pick up the scattering amplitudes around, let's say, the bamboo rod, and that sets up a sort of scattering characteristics according to the receiver angle as shown here by this in this time-lapse movie where the receiver picks up the scattered field. So this is only half of the let's say measurement effort that we have to take into account, because we want to go for a bistatic scattering or radar cross-section. That means we also have to rotate the bamboo stem accordingly, and in order and also to pick up the corresponding scattering characteristic, and that is done here within this let's say back and forth scanning of the receiver around the bamboo stem. We see on the lower left that we start complementing the data set of the bistatic scattering cross-section. So that will take some time. And at the end, we will also display these diagrams in a polar coordinate system, which is much more intuitive to understand. So that will happen in the very next step. What we will see then is here we see the, let's say, the scattering diagrams around the bamboo stem. And these measurements are then used in order to carry out the, let's say, inverse problem. The inverse problem uses our let's say, as our T-matrix approach. So the bamboo stem is parameterized by the inner and outer diameter and the eccentricity, and also the material properties of the dielectric function. And what you see here is this sort of ongoing optimization process, which will end up with the best fit to the measurement. And that would be also that would also provide us the, let's say, the reconstructed cross-section of the bamboo stem. So this looks pretty fast, but it's actually a slowed down version, because using our algorithm in real time, that would have been carried out within parts of a second. So moving further to the next section, insect and bee monitoring. So I always say insect and bee monitoring, but mainly we are looking to bees, because that was actually our starting point in insect monitoring. Nevertheless, there will be also some activities looking at mosquitoes. So insect monitoring, and in particular bee monitoring, the goal here is to use a millimeter wave terahertz FMCW MIMO radar system to monitor the plant pollinator pest interaction and also the behavior in a very local ecosystem. So you could think of my starting slide where you see the blossom and the hovering insect. That would be such kind of a local ecosystem. We could also use the same system at the entrance of a beehive to establish a sort of checkpoint that is capable to identify intruders like hornets or even varroa mites that are carried into the hive with the bees themselves. And more interesting, and this is also a more far-reaching goal, we also want to analyze aggregated mass flows at the entrance of the beehive, namely counting the bees. That's not that much an issue, but for example, also to look at the pollen that is transported into the beehive, and if it's possible, also the nectar by monitoring, let's say, the state of the digestive system of the bee, and that is something that might be pretty challenging. I will outline that a little bit later. So the differentiation here for the monitoring is based on radar cross-sections, radargrams, velocity vectors, and let's say the statistics of bee movement. Then the next topic here is actually bee exposure. So the goal here is to answer the question whether commonly known electromagnetic exposure limits provided by ICNIRP, based on the so-called specific absorption rates, and that have been, let's say, promoted for humans, and whether these are also applicable to insects and bees. So this is a very important question that I will answer also at the end of my talk. The second goal here is what are potential impacts on bee health and behavior? And this is something more for the future, because there we also would have to really closely interact with biologists, entomologists, and let's say also beekeepers, for example. So the analysis here in the exposure realm is based on computational microdosimetry using digital twins. So on the right-hand side, you see two joint publications from the group in Terahertz.nrw. In the first one, the older one, we just draw, let's say, our view on terahertz monitoring of insects and plants. And the newer one deals really with terahertz honeybee sensing, and lots of the results I show here are also coming from this paper. So let's start with the monitoring and the corresponding radar cross-section analysis that has been research that has been carried out in Jan Balzer's group. So they use artificial bees, meaning 3D printed bee mock-ups made of resin. So these are the very detailed bee models, because they are undergoing a stereolithographic additive processing. On the other hand, we have also, for example, the polymer PLA12, which is made with the fused diffusion modeling, which corresponds to the white bee here. And the question is, why at all using artificial bee or bee mock-ups at all? So that would be a real bee, but the difficulty here that real bees, dead bees, are really dried out. One could think of preserving them in an ethanol solution, which is usually done. And a soaked bee would probably mimic a living bee, but it dries out pretty fast, even during, let's say, a measurement. So one thing that we are also looking at here is using digital twins of B, homogeneous one and inhomogeneous ones, in order to also generate radar cross-section training data, because the simulation, as it turns out, are much faster than, let's say, the experimental data acquisition. So for the measurements, they use bistatic terahertz time domain systems in the frequency range between 100 gigahertz and 1.5 terahertz. Let's say for the generation of very precise reference cross sections and let's say radargrams. So what I show here in the lower part of the figure are radar cross sections, very detailed radar cross sections at 3 different frequencies for the green B mock-up. But one has to also say that they use 600 traces per angle are needed to resolve, let's say, this measurements, and that lasts for roughly 30 hours for 4 of such radar cross sections. So one has also to look at different faster measurement setups, and what they are using are FMCW radar system at 240 gigahertz with a large bandwidth of more than 50 gigahertz, and that has been developed in Niels Pohl's group at the Ruhr University in Bochum, and have been also provided for very fast radar cross-section estimating. So looking at the reconstruction from radargrams, the group of Jan Balzer has used a back projection algorithm based on this type of radargrams for the 2D reconstruction, and this procedure actually corresponds to inverse synthetic aperture radar imaging at these terahertz frequencies. And what we see here on the right-hand side in the figure are the red reconstructed shapes of the mock-ups. And we already see that the higher frequency, the B, is very nicely reconstructed. They are even capable to reconstruct a much smaller mosquito, as shown here. And but nevertheless, this let's say reconstructions takes actually a long time. So the reconstruction was successful, and that for it also validates, let's say, this reference radar cross-section data. But on the other hand, these radar cross sections are also simulated, as mentioned before, within an HFSS simulation platform in order to extend, let's say, the training data for artificial intelligence-based classification system. So to show also reconstruction for 240 gigahertz FMCW radar, they carried out a study. It is actually a model study or a simulation study to say, where they used the digital twin of a mosquito. They used the same back projection algorithm. And what is seen here on the right-hand side is a very nicely reconstructed mosquito showing the main features. With main features, I mean the wings, because it could be advantageous, and that is something that they are also looking into. It is to, let's say, also monitor the beat, frequency of the wings that might be also can be revealed from this type of FMC radar system. So moving now to a third measurement setup or a measurement system that would be a terahertz harmonic radar for bee tracking. The question is, why using harmonic radars? The harmonic radars allows a unique identification of, let's say, individual bees. So here I show a schematic. Let's say the bee to be identified would carry, let's say, a nonlinear tag. And this nonlinear tag might consist in a very simplistic way on an antenna and a nonlinearity that could be a simple diode, for example. And therefore, it would produce higher harmonics that are also detected then by the radar system. In this case, it's the second harmonic that we are looking at. And why using this kind of harmonic radar for bee tracking? Because harmonic radar tracking in radar entomology is not that new, it's already used. But in looking at terahertz frequencies, we can say that we will have a very small tag. That means also a very small payload for this individual bee. And this individual bee might therefore be also detectable even also within a scattering environment consisting also of other bees. But if we have a pretty small form factors, we can also go for a little bit more complicated radar tags, even active tags that helps us to maximize, let's say, the dynamic in the overall measurement system. Because a simple test structure operating at 50 gigahertz, they already shown you looking at the echo at 100 gigahertz for a target distance of 1 meter, we have to deal with 150 dB overall insertion loss of the overall system. So that's a pretty bold number that one has to deal with, and that puts also a challenge somehow to the sensitivity or the dynamic of the overall system. Something very interesting, what they are looking at is to use advanced optoelectronic realization of radar systems, allowing for a distributed and highly multistatic setting that could be, for example, be introduced in the inner surface of a beehive, where the transceivers or the transmitter and the receivers are connected via fiber optic, or they could also be introduced into a mating box as shown here in order to monitor, let's say, the queen bees. So that would really be a step forward in very precise insect or bee monitoring within the very small compound of a beehive. So let's move now to the last topic, namely insect and in particular bee exposure. And here I want to start with, let's say, a literature study provided by the Swiss Federal Office of the Environment. What they did 3 years ago, they looked into the body of literature, mainly or especially to covering 132 journal publications. And amongst different claims they made, they also have shown that let's say, the largest body of literature was, it's always dealing with an exposure scenario of various insects, was dealing mainly with a low frequency range up to 100 kilohertz. Then there is a considerable body of literature covering let's say, the frequency range from 100 gigahertz up to 100 kilohertz up to 6 gigahertz, and only 9 publications at this time have been targeted to really to the millimeter wave range up to 300 gigahertz. And they certainly say that the evidence level above 6 gigahertz is extremely weak, and they made also the claim there is an urgent need for research, let's say exposure research at these high frequencies. So that is one part of the motivation. The more important part of the motivation is that given the, let's say, modern mobile communication standards like 5G or let's say the future 6G, let's say the wavelength within these fields becomes comparable to the, let's say, extent of even small insects. And the conjecture here is that there is a possibility of a resonant electromagnetic energy uptake by this insect that has to be studied in a very detail. So that's the task we are looking at. And but that also means that we are looking to an organism with a very complex morphology. If you look at, for example, the honeybee as shown here, what you see here is a cross-section from a micro-CT image. And what is very impressive is the density of organic functionality. So here we have the head with the large visual cortex. Then here it's a brain ganglion for the olfactory system connected to the feelers. Then we have the muscle part, we have the digestive tract, we have the heart here. And all these functionalities are actually providing an extremely heterogeneous structure confined within a small volume. So that means in order to analyze that with respect to exposure, we have to set up a digital twin-based electromagnetic microdosimetry. So in order to get reliable digital twins, we also have to look at the real material properties of B components. And that is actually a very morbid endeavor. So Mendana Jalali of my group carried out this research by dissecting more than 100 dead bees. And what we see here are the material probes for the wings. This is the material probe for the cuticle. So the cuticle is the chitin armor surrounding any insect, and it is believed also that this chitin armor is slightly semiconducting. So that would provide actually a natural shielding against, let's say, electromagnetic fields, and that I will also show in a very instant whether this applies. We have a third material probe containing all the inner organs, and then also a material probe containing the overall bees. And these material probes are actually sandwiched between two clinch foils, which are then introduced within a transmission reflection measurement setup provided by Swiss 12. So here we see the probe and the measurement setup. And we are actually measuring the material parameters within the Ka band, let's say from 26 up to 40 gigahertz, and also in the D band, covering the range from 110 up to 170 gigahertz. So having these two, let's say, channels available, we want to reconstruct the dielectric function using a 5-pole Debye model, which is a pretty sophisticated model. But this model allows us to extrapolate, let's say, the dielectric functions up to frequencies of 500 gigahertz. So what I show here actually are the dielectric functions, namely the complex permittivity as a function of the frequency, here the real part and below the imaginary part. And just to point out, these, let's say, curves are according to the different B components. So looking at the wings, we see that the wings have a, let's say, nearly constant permittivity of 1. So they have the same permittivity as a surrounding air. And that was also one of the motivations why the B mock-ups do not have any wings, because that puts also severe challenge to the printing process. So just to look at the first exposure analysis using digital twins, what we see here is a 3D FDTD simulation where, let's say, a plane wave is impinging the bee from the frontal part and up to 40 gigahertz. We already see, we show here actually the electric field distribution. We see that the body of the bee, also the inner parts, are exposed pretty well. It's a homogeneous model here, so it's not mimicking the real bee in a certain sense, but we already can see that there is a sort of exposure moving towards higher frequencies. We see that the decreasing penetration depth kicks in, leaving the inner parts nearly unexposed. So that is also interesting. And what is really interesting is that we see, starting from 200 gigahertz up to 500 gigahertz, that the antennas, the legs, and the wings also start to contribute to the scattering cross-section. Okay, if we want to study potential effects, we can only do that using, let's say, a detailed digital twin that also mimics the anatomical parts or the compartments of the organs. And that is something that we are now currently very active in. So the main question that I want to answer here is, does the cuticle, this semiconducting chitin armor, has a shielding effect for the bee? And for that reason, we have set up a very simple model, just using the abdomen of the bee. The left column is, let's say, just a homogeneous bee model. And on the right-hand side, we see the bee model with the cuticle. And what can be seen is for lower frequencies, here at 6 gigahertz, we see a slight shielding effect, but the shielding effect vanishes if we move actually to higher frequencies. And this is due to the fact that, let's say, the displacement current through this cuticle, through the chitin armor, becomes predominant. So at higher frequency, we have to see that the inner parts might be, let's say, exposed. But looking at this, we also have to deal actually also with the material properties of the inner parts of the bee. So coming now to my last slide. and actually also the most important slide in this exposure topic. Here I want to discuss the energy uptake and the specific absorption rate in bees. The specific absorption rate is a very peculiar measure used in this bioelectromagnetic community because it measures, let's say, the power loss here against the mass density. And this peculiar figure can be easily linked to a temperature increase, which is actually that what might harm the insect or influence insect behavior. So what I show here is an illumination scenario according to P1, where the bee is illuminated from the side. And here these three bee models show the volumetric loss density at three different frequencies. At low frequency of 3 gigahertz, then an intermediate frequency at 25 gigahertz, and here the frequency 500 gigahertz. So at low frequencies, we see that, let's say, the loss density is really moderate and evenly distributed. Only the feelers pick up a little bit higher loss than looking at the 25 gigahertz. Here, this is an important wavelength because half of the wavelength is somehow corresponds to, let's say, the extent of the bee. So what we see here is a very prominent absorption in the head region, and we have to remember that the head contains all the sensory signal processing units in a certain sense. So here we really have to look into it very carefully. And then going to the very high frequencies, we see that the feelers are heavily exposed to let's say, this irradiation. And the feeler actually have a mechanical joint, and the mechanical joint is the ear of the bee. So that could be potentially also represent a hearing impairment for the bees, and that also has to be analyzed also in cooperation with entomologists. On the right-hand side, I show the specific absorption rates against frequency for the cuticle, where it has the largest, let's say, specific absorption rate, but that is not that much important because the layer is very thin. The most important figure is the, let's say, the whole bee specific absorption rate, taking into account the whole bee volume. Why is that the case? Because we could actually compare this figure with the ICNIRP exposure limit for a general public, also for human beings. And this limit, this so-called 10-gram SAR limit, is given at 2 watts per kilogram. So what we did, we extrapolated from this data let's say, the irradiation power density and calibrated our measurements to these kind of power densities and ended up at, let's say, a specific absorption rate of 7.6 watt per kilogram. I have to say it's a very unfair comparison, but at least it provides let's say, a very important fact that we really have to look into these figures because the figure is more than 3 times larger than that what would be allowed, let's say, for the humans. So coming to a summary and the conclusion. In conclusion, even if you have shown pretty nice and outstanding results, our interdisciplinary research on Terahertz plant and insect monitoring is still evolving. So somehow we are still carrying out engineering research. Let's say from an engineering viewpoint, the players in our ecosystems are quite challenging in terms of stochastic variations. That means variations in shape. Not every bee looks the same. Let's say bumblebees have much larger, let's say, shape dysmorphism. Then also morphology, then the constituents would also vary. And most important, also the movements vary. So what I show here on the right-hand side is a still of a very nice picture having a blossom of a plant. And here are the bees. If you would look at this picture in reality, you would see that this flower would bend stochastically around due to the measurement, due to the presence of the bee, and also due to the wind. So this is something that we really have to take somehow in account when we are talking about, let's say, a precise radar imaging of, let's say, hovering bees on a plant in this kind of ecosystem. So the radical miniaturization Terahertz engineering enables very close and hidden, so that is a statement that I already made before, view into the local ecosystem with an interesting possibility of what I would claim participant observation. So the term participant observation actually comes from ethnography, where sociologists dive into an alien society in order to carry out research on social interaction and so ever. So they are becoming native in this society. And let's say that the small form factors of our terahertz sensors and radar systems allow somehow for a similar approach. So we can introduce those tiny sensors also into a living society like a beehive in order to look at their social interactions, to say. So as an outlook, I just want to make a bold claim. We must collaborate way more closely with plant biologists, with entomologists, with neurobiologists, and ethologists, which are the behavioral scientists, and actually also with beekeepers. So thank you very much.
Thank you very much, Professor Erni. I see a couple of clapping hands in the room. Thank you, everyone, for attending, for your attention. I'd like to move on into our question and answer session. I wanted to start off on the topic of insects and bees. So we know that radar is already a well-established tool in entomology for remote sensing and tracking of insects. So why would you turn now to terahertz waves, and what advantages do they offer over traditional radar methods?
Yeah, I think let's start with why using terahertz waves on radar entomology, which is a well-established, let's say, tool for long-distance beam tracking. And the differentiation already is contained within long distance. What we want to look at is in really closed, small ecosystems. That is something at high resolution. So high resolution is something that we get out at terahertz waves on one hand, and on the other hand, these long-distance speed tracking radar systems are operated at pretty high energies, and that couldn't be used actually in let's say when looking very closely to such a, let's say, ecosystem. And I also want to address the last claim I made. The tininess of our systems allows us to really be in the midst of such an ecosystem, and that is not possible for present, let's say, for the traditional radar entomology, where they have huge parabolic antennas or radar antennas that points toward the sky in order to really look at the hovering bees or the flying bees. So for us, the task is really to be part of, let's say, this ecosystem, and therefore we need a high resolution on one hand, and also not that high, let's say, energy levels of the terahertz radiation.
Thank you, Professor Erni. I also noticed that your work has a strong emphasis on digital twins and 3D printed bee models.
Mm-hmm.
It was quite nice to see all the visuals in the talk today. But my question is, why not use real bees directly? And how confident are you that terahertz scattering from the models truly matches the real bees?
Yeah. It's a very important question, because that was actually the main concerns also that has been brought into our attention when we have submitted these two papers as shown in the talk. So why don't we use real bees? Let's say the reason is, I already mentioned it, dry bees dry out very quickly. So that the dead bees that we get from the beekeepers are, let's say, dried out. Let's say in order to somehow humidify the bee with ethanol doesn't work pretty well, because ethanol also evaporates pretty fast, and therefore we stick actually to mock-ups. And the question is really valid. So why have we so much trust in mock-ups compared, let's say, to the bees? There have been comparisons carried out, so the differences are not that large. That is a good sign. On the other hand, we are confronted with something that is common in this kind of research. So as engineers, we are, let's say, tackling with objects that are very random. They are even individuals in shapes and whatever. And we have to, let's say, generate knowledge that in a general sense. So to go for a mock-up is a very first step. And this might be a sort of pragmatism that could be also criticized. But on the other hand, it's one of the only possibilities to really go towards that direction. So I'm totally with you, claiming that one has, in an ideal world, to have mock-ups that really mimic the bee scattering behavior. But on the other hand, we are in a let's say, a very uncertain environment, and we have to really carry out reliable data. So the mock-up is actually a very good step forward into that direction.
How does terahertz sensing then fit into your broader long-term vision for plant and insect monitoring? And why do you think that this terahertz sensing is a critical part of that vision?
Yeah, terahertz sensing, as I mentioned before, allows us to monitor this very closely, these ecosystems. What we also want to do, and that is a long-term vision, let's say connected also to an application, what could think of having a sort of post which acts as a terahertz radar platform that can be introduced into let's say, a plant field oriented in the direction of a certain plant. So, and this would allow really a continuous 24/7 observation of this kind of plant, even during night. So this somehow includes monitoring systems based, let's say, on videos. So, and we think, due to the fact that the systems are so small, they can be easily introduced also in sort of tin poles that are standing in the very neighborhood of let's say, the plants to be observed or the insects to be observed. That is actually, from a systematic point of view, our picture that we have in mind if we are looking at the plant and bee monitoring, or at least the insect monitoring in an ecosystem out in the fields. The system that I described for looking into a beehive, that is something also on the long run. But what I also want to say, and that addresses also my last claim, we have still carried out our, let's say, ideas on what should be observed, but we really have to collaborate much more closely, for example, with entomologists, because they might also provide ideas what is worth monitoring, which, let's say, flight kinetics in what context should be observed. And we are very convinced that our systems can do that on a very high spatial resolution. But on the long term, I would say this observation pole for ecosystem is one thing. The other thing is this well-equipped, radar-equipped beehive. And then we also should really talk to the biologists, because I'm pretty sure that they have much more ideas where our technology could be applied to.
Thank you, Professor Erni. And one curiosity from the audience was about how close is this research to real-world applications in farming and agriculture, such as using them in the fields or at beekeepers' farms? And is there any industrial interest in adopting these sensing techniques?
Yeah, that's a very good question. So if you look at precise agronomy using similar techniques, we see that in agronomy, these systems are used more in an industrial sense. So they are looking at mass flows of crop, for example, where they detect the residuals of pesticides, for example. Or, as I mentioned, this Israeli example is already a sort of pinpoint technology on an exact plant in order to have a reliable real-time information about the plant. So what is the application in the industry? What I have to say here, we are still in a more scientific phase. I think from technology readiness level, the systems could be also applied in an industrial context, but we also have to prove that our very intimate view or a very close view on plants and insects have, besides the scientific, let's say, scenario, also industrial scenario. Let's say from the technology, I think that that's not a problem, but more from what should be observed, what is, let's say, the target of our curiosity. That might be, let's say, something that has to be proven also to the industry, but because industry uses this kind of technologies more in the industrial sense, and we are actually now looking more with a scientific angle to this kind of, let's say, biological organisms. Yeah.
Moving on to another topic, we know that video and acoustic systems are already in use to monitor closed ecosystems such as beehives or plant pollinator interactions. So why would you add terahertz monitoring? What advantages can these methods provide?
Yeah, let's say the acoustic system, they are really looking at the aggregated noise of bees. And the research there, it's also oriented according to the fact that, let's say, bee populations are challenged by environmental stress. And one important feature in bees are that the foragers that flies into the beehive, there is a sort of transfer of, let's say, nectar or whatever. So, and what is seen that there is, what has been observed, let's say from bee biology, is that the number of foragers compared to the numbers that are present at this, let's say, work interface within the beehive is not balanced anymore. That is something that has to do what I learned with this environmental stress, and that manifests with let's say an increased noise within the beehives. But it is an aggregated noise that just monitors the whole noise generation of the bee population. What we can do here is that we can also have a spatial monitoring of bees with what I've shown here also with the harmonic bee tracking. And during day of 24 hours. So one has really to imagine that the beehive is actually a pretty dark space. So there is no light within a beehive. And having a system that allows for monitoring at high spatial resolution is really something that would make an impact also for the entomologists.
I'd like to touch on the last topic, and that was plant monitoring. So you mentioned using terahertz waves to detect heavy metals in the plant leaves. What makes this approach appealing to plant biologists, especially since they already might have established methods for assessing heavy metal contamination?
Yeah, what I learned from the description provided by plant scientists is that if they want to measure the, let's say, the parameters of this type of contamination, they also have to dissect somehow the leaves. They also generate material probes for example, for inductive coupled optical emission tomography in order to look at the real content or the concentration of this heavy metal. So you always have to somehow destroy the probe. And our approach also allows, I want to also mention the research in the group of Niels Weimann, to actually clamp the sensors on the plant themselves, and the sensor will be present for the overall life cycle of this plant. So this is a total different approach that provides information, let's say real-time information of the living plant, and you do not have to dissolve that. So I have actually to also admit, in the case I described, the plant leaves have been prepared, so they have to be dried out, they have to be somehow pressed. So that is pretty similar to a biological approach used in biology. You also have to somehow prepare, let's say, the specimen to be ready for this, let's say, transmission setup. But I think We could also try to go towards a non-invasive method in order to mimic or monitor this kind of, let's say, heavy metal accumulation in plants.